Hello everyone and welcome to my channel Science Query. I am your host Dr. Asha, and in this video I am going to talk about pentose phosphate pathway. We are going to cover three important points. First, what is pentose phosphate pathway? Second, how it happens, and third, why this pathway is so important. A pentose phosphate pathway is a metabolic pathway for carbohydrate. It runs parallel to the glycolysis. It is a glucose oxidation pathway and it takes place in the cytosol of a cell. So from this definition we can see there are three main terms which is very important to describe. These are metabolic pathways, second important term is glycolysis, and the third important term is glucose oxidation pathway. A metabolic pathway can be simply defined as a series of chemical reactions that starts with a substrate and finish with an end product. And these pathways are controlled by enzyme catalyst. A simple example of a metabolic pathway is glycolysis. In the glycolysis metabolic pathway, the glucose is used as a start substrate. This glucose is obtained from carbohydrate and glycolysis happens in the cell. Glucose, with the help of various enzymes, undergo a series of chemical reactions forming many intermediate like glucose 6-phosphate, fructose 6-phosphate, and finally produce the end product, that is pyruvate and energy currencies like ATPs, which are used for other cell metabolic activities. Next important term is glucose oxidation pathway. It refers to the breakdown and processing of glucose into ATP and NADPH. Two important glucose oxidation pathways are glycolysis and pentose phosphate pathway. Now let us come to the pentose phosphate pathway and let us understand the pathway in detail. In our body, the carbohydrate is metabolized by these two different pathways, glycolysis and pentose phosphate pathway. Next question is that why we need two different pathways for the metabolism of carbohydrate? Well, carbohydrate metabolism through glycolysis produces ATP and carbohydrate metabolism through pentose phosphate pathway produces NADPH. Both the end products have different functions in the body, and based on the requirement, the cell will decide which pathway is to follow. Glucose enters into the cell through GLUT2 transport. In the cytosol of the cell, glucose converts into glucose 6-phosphate in the presence of enzyme glucokinase. In this process, one ATP is used and one ADP is released. Now this glucose 6-phosphate in the cytosol can choose either go to glycolysis or into the pentose phosphate pathway, depending upon the requirement of the cell. Glycolysis is a metabolic pathway that converts glucose 6-phosphate with the help of enzyme isomerase into fructose 6-phosphate. Then with the help of enzyme kinase and ATP, it converts into fructose 1,6-bisphosphate. Now this step is a key regulatory step and is also irreversible. Next step, aldolase cleaves the fructose 1,6-bisphosphate into two different carbon sugar, dihydroxyacetone phosphate, also known as DHAP, and glyceraldehyde 3-phosphate or G3P. The isomerase enzyme here rapidly convert the DHAP into G3P, ensuring both 3-carbon molecule proceeds through the next step. Then the glycolysis metabolism enters into the energy payoff steps, where the generation of ATP and final product pyruvate takes place. Pyruvate can be used as an intermediate for energy production. Glycolysis produces gross 4 ATPs, out of which 2 are used, hence net production in glycolysis is 2 ATPs. Now let us discuss about the pentose phosphate pathway in detail. There are two phases in the pentose phosphate pathway. First is the oxidative phase, Second is the non-oxidative phase. Oxidative phase has irreversible series of reactions. That is, glucose 6-phosphate oxidize into ribulose 5-phosphate and NADPH positive. Non-oxidative phase is a reversible series of reactions that interconvert various sugar phosphates. Like for the nucleotide synthesis, it converts ribulose 5-phosphate to ribose 5-phosphate and also rearrange the 5 carbon to produce intermediates that can re-enter into the glycolysis like fructose 6-phosphate or glyceraldehyde 3-phosphate. The first step of the pentose phosphate pathway is also a rate limiting step of the entire pathway. Here glucose 6-phosphate undergoes dehydrogenation reaction to produce 6-phosphogluconolactone with the help of enzyme glucose 6-phosphate dehydrogenase, and in this process a molecule of NADPH also produced. This enzyme glucose 6-phosphate dehydrogenase is a very important enzyme. It determines the fate of the glucose 6-phosphate, whether it will go to glycolysis or it will continue to pentose phosphate pathway. Glucose 6-phosphate is a 6-carbon molecule and it is converted into 6-phosphogluconolactone, that is also a 6-carbon molecule. In next step, the lactonase enzyme, that is 6-phosphoglucolactonase enzyme, hydrolyzes 6-phosphogluconolactone and convert it into 6-phosphogluconate. In this process, a hydrogen ion is released. The 6-phosphogluconate is also having 6 carbon. The next step is decarboxylation reaction step, where the molecule 6-phosphogluconate in presence of enzyme 6-phosphogluconate dehydrogenase converts into ribulose 5-phosphate, and in this process a second molecule of NADPH is produced, and since it is a decarboxylation reaction, therefore CO2 is released. This is the first phase of pentose phosphate pathway, also known as the oxidative phase, which is irreversible. Then the next phase, that is non-oxidative phase, will start. Here the sugar phosphate interconverts into each other and the intermediate can also re-enter into glycolysis whenever is needed. After the decarboxylation step, ribulose 5-phosphate is either used for nucleotide synthesis or it can enter into the non-oxidative reversible phase. In this phase, the ribulose 5-phosphate convert into ribose 5-phosphate with the help of enzyme isomerase, and the reaction is known as isomerization. Or it can convert into xylose 5-phosphate with the help of enzyme epimerase, and epimerization happens here. Both ribose 5-phosphate and xylose 5-phosphate are 5-carbon sugar. Let us just understand this reaction in a little detail. The ribulose 5-phosphate, which is a 5-carbon compound, isomerize with the help of isomerase enzyme and convert into ribose 5-phosphate. In ribose 5-phosphate, with the help of isomerase enzyme, the isomerization happens in carbon number 3. This ribose 5-phosphate can be used for DNA synthesis or RNA synthesis. Again, this ribulose 5-phosphate can also undergo epimerization reaction with the help of epimerase and it converts into xylose 5-phosphate. Both ribose 5-phosphate and xylose 5-phosphate are 5-carbon sugar and both are interconvertible with each other. Now let us go to the subsequent reactions, where again the ribose 5-phosphate, please ignore the spelling mistake, and xylose 5-phosphate will rearrange with the help of transcatolase enzyme, converts into glyceraldehyde 3-phosphate and pseudoheptulose 7-phosphate. This glyceraldehyde 3-phosphate is a 3-carbon compound and pseudoheptulose 7-phosphate is a 7-carbon compound. The transcatolase enzyme is a thiamine pyrophosphate dependent enzyme. Thiamine or vitamin B1 is an essential nutrient required by all the tissues including the brain. In its active form, thiamine pyrophosphate is an important coenzyme for several enzymes including the transcatolase. Now this glyceraldehyde 3-phosphate is having two options. Either it go back to the glycolysis cycle or it can continue further in the non-oxidative phase reaction of the pentose phosphate pathway. Next step is the 3-carbon transfer step. Here the enzyme trans-aldolase transfers a 3-carbon unit from pseudohaptulose 7-phosphate to the glyceraldehyde 3-phosphate produced in the previous step. This forms two more molecules, fructose 6-phosphate and erythrose 4-phosphate. Fructose 6-phosphate is having 6 carbon and erythrose 4-phosphate is having 4 carbon. This fructose 6-phosphate can enter into the glycolysis or it can continue the pentose phosphate pathway depending upon the requirement. In the final step of the non-oxidative phase of pentose phosphate pathway, the xylose 5-phosphate, that is the compound from the previous step, reacts with the erythrose 4-phosphate in the presence of enzyme transcatolase, and the transfer of 2 carbon happens here, which results in the production of glyceraldehyde 3-phosphate and fructose 6-phosphate. So this glyceraldehyde 3-phosphate and fructose 6-phosphate are again the glycolytic intermediates. Hence, they will go back into the glycolysis. Therefore, this phase is known as the non-oxidative phase. This is a reversible phase and involves the interconversion of various sugar phosphates. It allows the cell to adapt to its need. If the cell needs more nucleotides, it can convert the ribulose 5-phosphate into ribose 5-phosphate. And if the cell needs more ATP, it can use a series of reactions to convert 5-carbon sugar back into fructose 6-phosphate and glyceraldehyde 3-phosphate, both of which can re-enter the glycolysis. The key enzymes in this phase are transcatolase and trans-aldolase. The activity of pentose phosphate pathway is regulated by what cell needs, like whether it needs NADPH or nucleotides. The enzyme G6PD, that is glucose 6-phosphate dehydrogenase, is the rate limiting step of the pathway. It is allosterically inhibited by high level of NADH and stimulated by its substrate NADP positive. This feedback mechanism ensures that the pathway is active when the cell needs more NADPH. For example, during the periods of high fatty acid synthesis or oxidative stress, and is slowed down when enough NADPH is available. Next, let us see what happens if pentose phosphate pathway is absent in our body or cells. Genetic deficiencies of pentose phosphate pathways occurs relatively commonly in enzyme glucose 6-phosphate dehydrogenase. The G6PD, that is glucose 6-phosphate dehydrogenase enzyme deficiency, results in hemolytic anemia due to the red cell oxidation damage. But in severe cases, it also results in infections due to lack of leukocyte oxidative burst, highlighting the dual redox roles of the pathway in free radical production and detoxification. Well, that is all for the video of pentose phosphate pathway. If you like my video, then please subscribe my channel Science Query and please like, share and comment. Thank you.